Okay, well, welcome back. We're going to start with the last session for today. I can see the energy just emanating from you because you've gotten so much out of what's been happening so far. But of course, we've saved the most interesting part for the last part today. We're going to talk about clustering as one of the major tools. And of course, anybody who's ever heard of a microarray just knows that clustering was invented by Mike Eisen, Paul Spellman, Pat Brown, and David Botstein in this paper that they published in PNAS in 1998. And there's various ways that we know this is true. That paper now has close to 10,000 citations. It's one of the most cited papers in the last 20 years. And one of the ways that you can tell they must have invented clustering, if you look at the citations, the references at the back of that paper, there are 16 of them, 15 of which are to biologists, one is to a computer scientist, Cahonen for self-organizing maps. So clearly nobody had done anything with clustering before they published this paper. In spite of the fact that their erratum acknowledges that John Weinstein had published a paper the year before with colored, clustered heat maps. So I don't know, maybe John Weinstein invented clustering. Of course, these people are responsible for the standard color map that you see in heat maps. It's almost obligatory today that any paper that uses high throughput data has one of those red-green colored heat maps. They clearly didn't care that 10% of the adult male population is red-green colorblind. In any event, this well-known fact is clearly not true. Clustering was around long before they did this, but they are responsible for using red-green colors for the heat maps, just like this picture. If you're red-green colorblind, don't blame me, blame Mike Eisen. Okay. But this is what they look like. You've probably seen thousands of these. So part of what we're going to talk about is how do you produce them, where do they come from, and why? And if you look at the elements of this, right, typically there's a dendrogram, which is an all too fancy name for tree, across the top that clusters, in this case, samples in this example. There's a dendrogram along the left side, in this case, clustering genes. And there's usually the two-dimensional image that shows some expression of genes in samples on some scale that may or may not be fully explained. And it may include optional elements like this one did with a color bar across the top to indicate groups, or a color bar along the side to integrate other kinds of groups. The standard reference for this is it's called two-way clustering. Even though you don't actually do it in 2 dimensions all at once, you typically do 2 different one-dimensional clusterings and glue them together around the heat map. Okay, so we know that we're going to build trees. The standard way to do this is called agglomerative clustering. That's a big, hard-to-pronounce word for bottom-up. You start as though all of your things you want to cluster are thrown on the ground, and you pick up the 2 that are closest together and mark that, and then you keep picking others up and build your tree up from the bottom. But of course, there's some questions about how that's going to work. How do you define closest? All right, how do I tell the distance between 2 individual objects? Mathematically, they're vectors in whatever space we're looking at. How do we define the distance? And then you have to realize there's a second question you should ask, because partway up, you now have not just individual objects, but you have clusters, and you have to say how far apart are the clusters. So there's 2 different steps where you have to give a definition of distance before you can even start building the dendrograms that you use on the heat map. Whenever you see one of these diagrams, you should check the method section, see if they tell you how they define the distances. If they don't, then they're probably hiding something, or they're incompetent. They don't know that there's lots of choices here that they must have made along the way. Okay, how do I know that? Well, what I have here are 9 different dendrograms on exactly the same data. This is the data that was shown on the left-hand side in the initial heat map. And the colors here are the same as the color bar I used in that first heat map. And you can tell them because they're the colors in the top left panel. Right? There's orange, green, blue, brown, gray, red, purple. Okay? That's the order they were before. This is all the same data. The different rows are different linkage rules. Linkage rules are how you tell the difference between groups. The different columns are different measures of distance. And what I want you to get out of this is that Every single one of these dendrograms is different. The choices you make about what is the distance measure and what is the linkage rule changes the structure of what you get. So that's why you need to ask what people are there. And of course, we're going to use these to do biology. So the right question to ask is, well, if they're all different, how do I tell which one is really the truth about the biology? I'm not going to answer that question for you. I'm going to give you some tools for how to think about it, but there is no good answer to it. Nobody knows which of these methods truly reflects biology, and it may be different in different contexts. Okay? So clustering to produce dendrograms, all those algorithms always produce dendrograms. They always produce clusters whether they're real or not. Okay. So we're now going to try to start digging into this a little bit deeper so we can explain what these concepts are, right? In some vague sense, again, what we want to do to find clusters is to determine which things are closer together and form, in some sense, more coherent pieces, rather than just one unstructured clump. As I said, we need to define distance. And anybody who's gone through high school successfully knows at least one measure of distance because they probably beat you over the head at some point in a geometry class with Euclidean distance, right? And I've written in R language 2 forms for this. I could put the mathematical formula here, and half of you would then immediately stop paying attention. I'll put some formulas in later. I want a few of you to keep going at that point. But the general idea, right, is that you take the difference between the vectors of data, square those, sum the squares, take the square root. That's that definition of Euclidean distance in the little yellow box partway down the slide. And you can also write this using the dist function in R. We don't care. That's the first measure of distance that you should think about. Okay? May or may not be right, but that's the first one that we have. That's a definition in gene space of distance. But there's a lot of others that are already built into R that people have considered in various contexts. You would probably be amazed to learn that if you're just measuring distance between binary vectors, so a vector that just has zeros and ones in it, there's a paper that lists 76 different measures of distance between binary vectors that they looked from other papers in the literature, because this was the review article. There's lots of ways to define distance. Some of them, well, maximum is what it means. You know, that's, if I'm looking at vectors x and y, find the biggest value between them and forget about all the others. There's also a minimum of that. There's one called Manhattan distance, which, unlike the Manhattan plots that Sheely Lin did yesterday, is motivated by the fact that there are these nice rectangular blocks, and Manhattan distance is how many blocks you have to walk to get between two places. Okay. Not sure why Canberra distance is named after that. I don't think it has to do with the shape of the city. I think it has to do with that's where the statistician who came up with it was housed at the time. Anyway, there are a lot of these. In red, I flagged the one that is used most commonly at present in applications to biology, which is a measure of distance based on Pearson correlation. You want things that are highly correlated to be close together. And the maximum value for a correlation is 1, goes from -1 to 1. So if you had a +1 correlation, that formula would subtract it off from 1, so it would give you 0 distance. So perfectly correlated is 0 distance apart. Perfectly anti-correlated is as far away as you can get. That's a distance of 1 in this normalization. There's variants of that. You can take the absolute value of the correlation. You can take the Spearman rank correlation, which is how correlated are the ranks as opposed to the observed values. But there's lots of different distance measures. And there's lots of different linkage rules. And I can illustrate the first 2 of these. If this is cluster 1, see we're on video here, and this is cluster 2, and we say how far apart are they? Single linkage takes the closest things, that's my thumbs, and says the distance between my thumbs represents the distance between the groups. Complete linkage says which ones are furthest apart, and that's the distance, so that would be the distance between my pinkies if this is. And average linkage, well, there are various notions of average, but you average over all the distances between all the things in the groups. And you can do that with a mean, you can do that with a median, or you can first average the items, if that makes sense, and take the distance between the averages. That's known as centroid linkage. And I've added one here that looks very different from the others. It's the only one that has some sort of statistical motivation, which is that with Ward's linkage, the rule is to not so much look at distances, but to look at some measure of how poorly you would classify things if you put things together. And that's measured by the within cluster variance, the within cluster sum of square distances. And you want to join things in a way that results in the smallest within cluster variances, given that you have to put something together in the things that you're clustering. Okay, so we're going to try to now actually look at this in practice. That's sort of as much of the theory of clustering we're going to look at for the moment. So we want to do this in R. And as I've already told you, every clustering algorithm guarantees that it will produce clusters. Doesn't guarantee truth, doesn't guarantee the right clusters. You always get clusters out. So what I like to do when something claims to be able to do that is test it by saying, let me simulate something that has no structure and see how much structure the method pretends to impose that isn't actually there. So that's what we're going to do. This block of R code here says I'm going to simulate something that has 1,000 pretend genes and 50 pretend samples, fill up a matrix by putting in data randomly selected from a normal distribution, so all the data is the same, and see what happens when I try to cluster it. So here's the first example. This one uses Euclidean distance, and it uses average linkage. We see that d-euc to remind me that it's Euclidean in the first line, and I see an average in the method for how I cluster it in R. And here's what a typical average linkage clustering looks like when there's no structure. And you probably believe there's no structure. Okay, I don't see any big stuff other than sort of singletons hanging off the ends that matters here. And again, this is my random normal data. And here's the same data clustered with single linkage, where you're even more sure that there's no clusters, but it's often the case that single linkage produces this kind of string-like structure, sort of attach one extra thing along the end of the string as you go. This, of course, is why nobody in applications uses single linkage for anything, because you can't find anything. Doesn't matter whether it's true or not, you have to find something or you can't publish the paper. Okay. So here's complete linkage, which, again, there is no structure here. This is the same random normal data. Complete linkage, again, Euclidean distance, the same set of samples, produces something that begins to hint at structure, right? Somebody could probably, if they were slick talking enough, convince you that there were a couple of clusters here, maybe even 3. Okay? Clearly, you can just see it. You can see those groups coming off at a high level there. This isn't truth. There is no structure here. So one of the things we're going to look at is how do we detect that there is no structure here? Okay? And just to convince you that it isn't completely just the distance measure here, here's complete linkage with correlation as our measure of distance, measure of similarity. Here, you might even be more convinced that there were 3 groups than if you used Euclidean distance. But the true number is there's only one clump. These are all the same data. Okay, so one of the things to keep in mind about these dendrograms, one of the things you should ask, so what part is invariant under this? I should have put an example here, but sort of the underlying theme to this, at least to the beginning, is that you should assume that all dendrograms are lying to you until they convince you otherwise. And there are ways that you can make it look like lying even more. So if I go back here, there's a linearity in the display. This just goes from left to right across here. That's meaningless. You have to think of this as a snapshot of a Calder mobile. Any of those branches you can flip around. And so at the linear level, you can make things look close together that aren't there. I can point to examples of papers where they've said, oh, look, this group of samples all clusters together, and when you trace it up, it's as though they flipped it, so they say, this is one group, and this is that same group, and they're claiming they're close together. But they're not, right? You have to look all the way up the tree to see that they're actually on completely different branches. So that's one of the things that you're allowed to do. You can flip any single vertex here and move stuff around and change the one-dimensional, apparent one-dimensional structure. Okay? And so given that, it's important to think about, in terms of hierarchical clustering, what I mean by a cluster. And that's, if you clip something off at a branch, everything below that is a cluster. So I've put a red box around a thing that I would call a cluster. And if I clip lower, I might put the red box around something different. But everything that's below a place where you would cut off a branch is what's potentially a cluster. Not a meaningful cluster, not a real cluster, not a piece of biology, but a cluster. Okay, so now now that we know how to make dendrograms out of meaningless data, now we're going to make entire heat maps out of meaningless data and see what we can get there. So we're actually going to delve into how you do this in R briefly. And you do that using a function cleverly called heat map. So you just take your data matrix, feed it into heat map, and what comes out is a cluster. And depending on the resolution of your screen, sometimes you can see the entire dendrogram and sometimes you can't. There is a dendrogram on the left-hand side where most of the lines have been clipped off because they're too thin for where the pixels are. We'll live with that. That's not the ideal way to do it. First of all, right, it's not the traditional red-green Eisen color map, right? It's a color map that R uses by default for all images, known as heat colors. You will also note there is no scale there that says what's low and high. Everybody knows that you start at red heat and then go up through yellow to white. That's the scale. You should probably put the scale on it at some point. But nobody's used to seeing these other than the people who developed R originally 20 years ago. So we're going to delve into the heat map to try to figure out how to make better heat maps. This slide is here to show you that from a computer science perspective, the heat map function in R may be a contender for the worst designed function in history. Why? Well, nobody can keep in mind more than 7 things at once. There are actually psychological studies at this, how many things you can remember. And if you sort of think, how many arguments does this have? How many different things? Well, I think there's about 20, but it's hard to tell. You have to count how many commas are in there to break things up. So there's all these different things you can tweak in the heat map. Okay? So it's really hard to figure out how to use it. So I'll tell you how I use it as we sort of build up some of the pieces. So one of the things that I'm going to do is put in the Eisen red-green heat map instead of that garish yellow-red one. Okay? Now one of the things that you see here is that by default, the colors look really boring. They're sort of washed out. Well, that's true because there's no structure here. That's part of it. But we can still enhance that and bring out a little bit more color, and I'll show why. By default, the color scale is determined by the highest expressing one being the brightest red, and the lowest expressing one being the brightest green. And there's probably like a single really bright green pixel and a single really bright red pixel. And those are the outliers on the normal distribution, and most of the stuff is near the black in the middle. Okay, so you probably don't want to do that with heat maps. And you probably want control over how you cluster things. So instead of letting heat... Oh, I forgot to mention that. Let's go back a moment. Notice we have no idea how this got clustered. Dendrograms got built. Mistakes got made. Okay? Turns out that by default, heat map uses Euclidean distance and average linkage, but unless you read the manual pages, you don't know that. Okay? And if you're a control freak and you want to control that, you can actually cluster things separately and feed them into the heat map. I would say use my clusters, not yours. And you probably should do that. So I'm going to cluster both the samples. Remember the Unix influence on our SC is my name for the sample clusters, and GC, surprisingly, is the name for gene clusters. And the distance, if you look at the difference here, right, we have hclust, which is a general clustering algorithm, and you use some distances. And here the difference is, this is on the data matrix, and this is T of the data matrix. That's the transpose. So that's why you're doing different things being clustered. And in both cases, oops, I didn't want to click that. In both cases, I'm using Pearson correlation as my measure of distance, and I'm using Ward's linkage. So I've got the documentation there that I'm using those instead of Euclidean distance or average linkage. And I get to feed both of those into the heat map function as the arguments rho v and col v. I'm not even sure what the v stands for there. I assume vector, but I don't know. And so I convert those clusters into dendrograms, feed them in, and my heat map will use my clusters instead of theirs. And the resolution on this machine is really annoying because my dendrograms are all broken up in this. But in any event, they are different than they were before. Okay, so that's the first part, is I can control the clustering algorithms. But to control the colors, right, I told you that it's set by default by whatever the highest value, whatever the lowest value is, and those are often outliers. So one of the ways to deal with that is to do two things. One is to rescale the gene data so that it's on a level of standard deviations. That's what the scale function does. It makes each gene have mean 0 and standard deviation 1. And then truncate, so you're looking at a certain number of standard deviations. So the things that are outliers are all going to be truncated and not, you have an outlier that's 9 standard deviations and everything else is between -3 and 3, you're not going to throw off your color scale. So that's what this does. It scales and truncates the data. Notice after clustering. So you're clustering on the real data. You're changing the data for display purposes only. Okay? And so you put this in. We've put in our dendrograms here. We've got the red-green heat map. You tell, I also didn't tell you, heat map by default has done some rescaling that it doesn't tell you about until you know it. This is, don't scale it. I've done that myself. And make the data go between the limits I've set. If I've truncated, it's possible that I set limits that didn't actually truncate, and something might be a little bit lower. This makes the scale be symmetric about 0. It goes just as high in one direction, just as low in the other direction. And if you do that, well, you can make the colors a lot brighter. It's still the same lack of structure, but it's perhaps more interesting artistically at this point. Okay? So we can control the clustering, we can control the colors mostly by scaling and truncating. And you can also change the aspect ratio. The built-in heat map function by default believes every picture should be square, and so it does that. There's a variant inside the class discovery package that I wrote because I didn't like them all square. If I have a lot more genes than samples, I want to be able to make it fit better on the screen this way. So you can control the aspect ratio as well. Okay, so what do we know? We now know that even if there's no structure, we can make pretty heat maps. But what we'd like to answer is the question of when do these clusters have a chance of meaning something? How do we tell? Okay? So there are different ways to try to do this. I'm going to talk about two of them. The first one is what I call Bootstrap Resampling. Bootstrap is a general statistical technique, I think introduced originally by Brad Efron at Stanford, who's worked out the details of it in some context. But the idea is to resample the data. So if my columns are samples in what I want to do, and that's what I want to cluster, and my rows are genes, what I'm going to do is make another matrix out of my old one by randomly sampling rows from my original data to put in this new data frame. And I sample with replacement. What that means is I could sample one row accidentally 10 times and leave out a bunch of other rows. So what I'm doing with this resampling, this bootstrap sampling, is I'm changing in some random ways the weights given to some of the genes. And I can do that a whole bunch of times. So if I make this gene more important and others less important, how does the clustering change? So there are other ways to perturb the data. This is the simplest one. You can also perturb it by adding noise to the matrix and reclustering. But in either case, what you're going to do is you're going to recluster many times and ask, how often do things go together? So again, this class discovery package, one of the ones we had you install yesterday morning, has the tools to do this. It has a function called Bootstrap Cluster Test. And you put in the same data matrix you've been using. In this case, that's one with no structure. Cut hclust that's some description of what function you're using to do your cutting into pieces. We're going to cluster it so that we're going to say there's 4 groups to where we cut off the dendrogram. We're going to use average linkage, Pearson distance, and we're going to do this 200 times. And then we're going to draw an image of the result. And I decided not to use the red-green color map because I don't want to confuse this with the original data. This is some measure of how often pairs of samples get put into the same cluster. The scale here goes from blue, pure blue at 0, to pure yellow at 100%. So stuff that's yellow always gets put in the same cluster. Here's the trick question. Why is the diagonal yellow? That's the diagonal. Everything is in the same cluster as itself. So we know what perfect clustering is. Why is everything else mostly blue? Because it's random data. There is no structure. So this test is actually showing us there's nothing that consistently goes in the same cluster as something else except itself. So just this bootstrap reclustering, most of the time you're shuffling everything around and not keeping 2 things in the same cluster. So that's why this cluster test is useful. There's this nice visual feedback that says, in this particular case, there is no structure. As I said, the notes are all there. Some of these are very wordy slides so that you can go back and read them later. And many of those I'm going to skip over quickly in my presentation. Okay, this just says there's some other options that we can do to that. Now I want to show you my next favorite trick for how to lie with clustering, which is to first filter on the genes in a way that keeps track of an outcome that you would like to emphasize. So the idea here, right, there are many papers that in fact have figures like this. They have 2 groups of interest. They do differential expression, all that stuff that Greg Rampala and Maciej Peszak were doing in the last 2 talks. So they find the list of genes that are different between group A and group B. And then they do clustering just using those genes. And then they say, look, when I cluster, I can tell A from B. That shouldn't be surprising. If you picked things that could tell A from B and you clustered and couldn't tell them apart, that would be surprising and would be worth writing about, except that people would say your data is crap, and so they wouldn't accept the paper. Okay? All right, so I did this on my simulated data set. I just arbitrarily said the first samples, 1 through 25, are group 1. The last samples, 26 through 50, are group 2. And I did t-tests on my 1,000 fake genes and picked the 50 best ones to tell those apart. And I just clustered with those 50. And if you look carefully, in the left-hand branch, most of the numbers are less than 25. 1, 3, 5, 17, etc. And if you look at the right branch, most of them are bigger than 25. So this looks like I can tell the first 25 from the last 25 on something that has no real structure. So you can see how you might use this to lie with clustering. Okay? The bootstrap doesn't save us. If you do the bootstrap just with the 50 pre-selected genes, it says that structure's real. There's 2 groups. The red ones are the first 25, the green ones on the bars are the second 25, and you can mostly tell them apart, and they mostly get stuck in the same groups. That's because you've already peeked at the data and picked out the genes that tell red from green. And so even if you bootstrap it without bringing the other genes in, you can't tell them apart. So you have to be very careful. If you're going to do any of these kinds of clusterings, you should probably do them, if you filter genes at all, with something that doesn't really directly relate to the outcome you want to show. Okay? You could filter on total variation. These genes are the most variable in the data. That doesn't mean they tell group A from group B. You could filter on I think chromosome 15 is important in this disease. I only take the genes on chromosome 15. Or we could say, I think apoptosis is important. I'll take all the genes involved in apoptosis. So criteria like that that don't directly reflect the categories you want are fair to use. But if you first do this filtering on stuff that wants to highlight a particular thing, clustering isn't unsupervised anymore. You've now supervised it by the choice of things you use to do the clustering. Okay, so having simulated nothing to prove to you that I can lie as much as I want with clustering, we're now going to try to see what we can do if we simulate something. So here's a small simulated data set where I've colored the 5 different groups, right? We have blue in the lower left-hand corner, we have magenta at the top, we have cyan in the lower right-hand corner, and we have red and green sort of overlapping because I only get 2 dimensions to show here. This is actually higher dimensions than that. So red and green really are different, but maybe not as different as blue, cyan, and magenta. And if you cluster this, well, I forgot what the map is between A through E and the colors. I probably should have been more careful about that. But if you look carefully, you can see that there are 3 groups that you can pretty much tell apart here. There's a bunch of C's on the left, a bunch of D's on the right, a few B's sort of to the right center, and the A's and E's are a little harder to tell apart. So I guess the A's and E's are the reds and greens, and the others we can count how many they are to tell which they are. So just doing clustering on this, we're pretty much okay. And if you bootstrap this, right, you pretty much see, oh, what do we see? 3 groups, 5, there's supposed to be 5 groups. Well, there's sort of 3 for sure, at least that we can tell, right? That cyan group we can see is pretty solidly a block of yellow that goes together without anybody else mixing in. The green group, actually, surprisingly enough, is fairly consistent. And it's the red, blue. I bet these colors are different. There wasn't a yellow there. Damn. But anyway, there's another 3 that are a little harder to tell apart. Although, if you take that same piece and reorder it by the samples, instead of reclustering it based on how often they go together, you can sort of see hints of a block here that's consistent, a block here that's consistent. Here's one that sometimes goes with this group or with this group. So you can sort of see the overlays there. It maybe tells you there are 5 if you know what's going on there. So it's a little harder. Once there's real structure, the bootstrap tells you that there's real structure, but maybe harder to say how much there is. Okay, so we're now going to take a deep breath and think about what clustering means slightly more abstractly. And I know the general rule, because of this equation, half of you are now completely lost. Okay? But we're going to try to get through it anyway. We're going to look at a different notion of clustering. Hierarchical clustering, things that build trees, are not the only way to cluster samples. And in a completely abstract sense, clustering is sticking labels on things. So if I think there are 5 groups, I have 5 kinds of labels, and each sample gets a label on. So that's what I'm going to think of as clustering for a moment. And there's a method known as k-means, where you have to specify k up front. How many groups do you want to find? And k-means will do that, right? So you specify the data matrix and the number of clusters k. And what's supposed to come out is this cluster assignment. For each object, you get a class, which is a number from 1 to k. An integer from 1 to k. And the idea, as I said, is to minimize the within cluster sum of squares. So what does that mean? What is this gobbledygook? This is sum over all samples. This is sum over, over all classes. C is a class, so we're supposed to get k class. So for each class, and for each pair of elements in that class, compute the square of the Euclidean distance. Okay? So how much variance is there within a class? So you want to make that as small as possible. That's the precise measure of what you're trying to do. Trying to clump classes together if there's small variability within classes and maybe large variability between classes. Okay? This just looks at the within classes. And the algorithm is you make a random guess at what the centers could be. So you pick If k is 5, you pick 5 potential centers of classes. Then you go through for each data point and you say, which center is closest? Put that label on it. And then after you've assigned all of those that way, for each thing that's got label 1 on, you average them to find the center of your group 1. Now everything with a label 2, you find the center there. And now you have new centers, so you repeat this. And you keep repeating until you get tired or until you think your algorithm isn't going to do anything better. So the usual description is you repeat until the assignments stop changing. In most actual implementations, it's repeat up to 100 times or until they stop changing, because otherwise you could go on forever and your computer would be in an infinite loop, and that's a bad thing. But that's basically the idea. And we can look at what happens here. So in this plot, the colors have changed because the thing you called number 1 in this labeling may not be what was called number 1 before, so the colors might change. But in fact, this particular algorithm got all of the samples correctly in the group they belong with. And there are little circles here, open circles. You have to look closely. There's one of them here, one here, one here. Those were the random centers. And the X's is where the final centers are. So that turned out to be the center of the cyan group. That turned out to be the center of the blue group, the magenta group. So this looks like k-means is a great clustering algorithm. The dendrogram didn't look like it got them all right, but k-means got them all right. You might note the take 1 in the title of the slide. Here's take 2. Ran k-means again, chose different centers, and it did a much worse job of clustering. It gets the bottom left-hand corner group right. Looks like it gets the magenta group right. But the other 3, well, this X for what's now called green has now split things up into 2 places. It's made a mess of things because it got a bad set of starting points. Here's the 3rd time I ran it. And this literally was the first 3 times I did it. I just got lucky that they were in this order. This one does even worse. It can't even get the cyan group that's the easiest to tell apart because it picked 2 centers in there and it made 2 groups out of that. Well, that sounds terrible. How do we fix that? Well, what that said was that the problem is the starting points. If you get lousy guesses for the starting points, you get lousy conclusions. But maybe you can tell which one is best overall. So our measure for best overall is supposed to be, from the algorithm, the within cluster sum of squares. And the implementation of k-means in R gives that back. So for these 3 cases, you can look at what those values are. Okay, and we see that the first one at 25,706 is better than the second one at 25,736, which is better than the third one. So we actually know that the first one is better. What we've also discovered here is a fundamental fact about algorithms that try to optimize something. Right? This is minimizing. It's a kind of optimizing. Most algorithms to optimize things rely on heuristics rather than doing a complete search. What this means is there's a chance it will converge to a local optimum instead of the global optimum. That's what happens with k-means. It's not the only algorithm that does that. For anything that doesn't exhaustively search, runs into that problem is it can get stuck at local minima or local maxima instead of at global minima or global maxima. So one of the ways to fix that, at least in k-means, is don't do it once. Do it a bunch of times and pick the best one. That doesn't guarantee that you get the global maximum, but it gives you a much better chance. So there's a function, again, in the class discovery package called repeated k-means that tells you how many times to do it. And that's the implementation. Don't worry about it. Okay? Now, that formula that I put up there that I know scared some people off had Euclidean distance built into it. Somebody soon realized that you could do the same thing with any old distance, and they called the method partitioning around medoids, or PAM. you get an idea of when the book was written that has this method in it, is that they describe a series of different clustering algorithms which are called Agnes, Daisy, Clara, Pam. I've forgotten the others. I don't think anybody would quite do that anymore. They came up with acronyms that could spell out female names. In any event, what's different about the formula here, you note that it's not as scary because one of the summations has disappeared. That was the summation that explicitly put the Euclidean distance. Here you put in whatever your favorite measure of distance is instead, however you compute it. Now they realized when they did this that the problem in the original algorithm was the averaging step. That's where that stupid word medoids comes from. Because if you don't know what the measure of distance does, you don't know how to find the average of a bunch of answers. And so what they do instead is, right, so you make a guess, you assign each point to its closest center, and then instead of doing averaging, within each cluster you find the one that minimizes the distance to everybody else. So you find the particular example that you actually had that is closest to the other members of its group. And then you run the same kind of algorithm. To do that in R, you load the cluster package, you call Pam, it gives a result, which turns out to be terrible on this data set. And that's because of the random choice of centers. Unfortunately, there's no easy way to fix this implementation in R because they've hard-coded inside the algorithm how they pick the centers, and they don't let you feed in a different set. So we're not going to use that, at least here. But in principle, we could do this with correlation distance or Canberra distance or any of those other distance measures. Okay? Now, neither k-means nor Pam nor hierarchical clustering answers the other question that's sort of been floating through this, which is how many clusters are there really? Okay? In order to run k-means, you have to tell it what k is. You have to say, I believe there are 5 clusters. If somebody else believes they are 7 clusters, they're going to get a different answer back. Okay? And why you should believe that, I don't know. From what we've done so far, you don't know either, but we're going to at least try to get at this. Okay, so how do we tell which k is best? And I'm going to describe one measure of that. Well, there's another formula here. This is an idea known as the silhouette width. Okay, so the silhouette width is a measure for each object that you're trying to cluster of how much you believe you put that object in the right cluster. And so you measure 2 different things. One is you say, how far on average is this guy from everybody else in its cluster? But then you also, for each other cluster, you say, so what's the cluster that's second closest to this, and what's its average distance to those, right? Is this somebody who's like, on the border of his cluster and maybe looks closer to the guys in the next neighborhood instead of in this neighborhood? So A of I is how far am I to the guys who have the same label as me. B of I is how far am I on average from the next closest group. And then there's this funny formula. You take the difference between B minus A and weight it by the maximum of A's and B's. God, what does that mean? Well, we're going to figure out what that means so that we can see how to interpret this, right? So if you're really where you're supposed to be, A should be much bigger than B, right? So what does that mean? Oh, sorry, A should be much smaller than B if you're in the right cluster. So B should be the biggest. So this is B minus A over B. So that's 1 minus something, right? And the smaller A is, the closer you are to your own people, the closer you are to 1. So a value of 1 or a value near 1 means I'm very sure you're in the right group. On the other hand, if you're clearly in the wrong group, B will be smaller than A, so that'll be B minus A over A, so that's something minus 1, and so you'd be close to minus 1. Any negative value, you think you're closer to some other group. And if you're close to 0, you're sort of on the border. So that's the way to think about this, that maximum is just scaling things down so everything you get is on the same range. Right? So it's some measure. How much do I believe that this individual guy is in the right group? And then their argument is you use the mean silhouette width to tell which is the right answer. So if I say that there are 2 clusters on my simulated data with 5 groups. What do you see? Well, cluster 1, a lot of people look good, and these guys are negative. In cluster 2, there are a lot of guys negative, and there's an average silhouette width of 0.02, whatever that means. So we're going to flip through a bunch of these. Here's 3. Groups 2 and 3 have some guys that are clearly wrong. We had a 0.03 for the silhouette width. Here's 4 groups, 5 groups. That's the truth, by the way. 6, 7. And here's the actual silhouette widths, which don't actually change all that much. They go from 0.19 to 0.027, 029, 028. So the biggest one in this case is 4. But it's only a little bigger than 5. So if you're strict about this, this says there are 4 groups, which isn't quite the right answer. But you could maybe argue that there are 4 or 5 groups depending on how much you believe the significance of the difference between 0.0287 and 0.0290 really is. Okay? So it gets close to the right answer here. You can also do this with other clusterings, so I can apply it not just to PAM, which is what was there. I can compute silhouette widths out of, for example, the k-means clustering I did. It actually has not quite as good a score, even though it does fairly well. And if you plot the silhouette widths, though, for the k-means clustering, even though the average may be close but not quite bigger, you see that there is nobody with a negative value here. So it tells you that it believes that everybody is in the right cluster, despite of the fact that the mean being wrong. You can also go back. Remember, we did hierarchical clustering of the samples early on. Okay? And so in order to get things right, there were a few isolated ones. You have to break it up into 8 pieces to get all 5 groups right. Maybe not know that, but OK. And I get a score of 0.08 that's a lot bigger than the 0.02 somethings that I saw. Why do you suppose that is? Well, if I show you the silhouette width plot and say, why do you suppose the average here is so big? It's because 3 of the groups only have 1 item in. And so the silhouette width for that 1 item is, if it's in a group by itself, it's clearly close to everybody in its group. And so it gets a score of 1. What this tells you is that the mean is a really lousy measure if you had singleton groups. You should probably use the median and not the mean. Okay? This is, again, a big block of words. All this says there's another way to do this called the gap statistic. In fact, there are about 30 different ways that people have, at least 30 different ways that people have for trying to get the number of clusters. Sometimes they agree, sometimes they don't. The gap statistic is a little more sophisticated. We're not going to use it here. I am going to spend a couple of minutes saying briefly something about principal components, which is a different way to sort of start discovering things in data. It doesn't directly do clustering, but it gives you nice 2- and 3-dimensional pictures. The idea of principal components, I have data in some big dimensional space. And the first question that principal component asks is, if I'm standing sort of in the middle and looking, in which direction do I look to see the biggest variability? In this room, if I'm looking around and say, which is the furthest away, it's right back there. Right? This room is furthest away from me that way. That would be the principal component. Okay? If I'm thinking of the data in some sort of semi-spheroidal stuff, the example should be a football. The principal component is the main axis of the football. And then there's a second direction to that, which is what mathematicians do all the time. Say, OK, I've solved the first direction. Now, perpendicular to that in all the directions I could look, which is the next biggest source of variation. So if you pick these out and then just use those principal components, use those directions as your new coordinates and stop at 2 or 3, then you get the best low-dimensional picture of your data that does the best job, in some sense, of keeping track of the high-dimensional structure. And so we can do this. There's a function called samplepca, again in the class discovery package that'll do this, and plot stuff. My original plot came that way. You can do it with other distance measures, and what else do I want to say? And you can show it in 3 dimensions if you want. We're going to come back to see how to do this, because what I want to do at this point, right, everything I've done so far is on simulated data. We're now going to go to the hands-on part and try to actually figure out how to do this on a real data set and see what we can learn. Okay, so I'm going to switch out of this, give you a chance to ask some questions while I get the next piece set up. One of the things I mentioned at lunch, for those of you who really want to follow on, is that you can go to the hands-on directory, coom/hands-on, and copy everything over. If you do that, you should make sure that you have everything in the same directory, just like I have it here. Okay, that will make things easier. And if you already have RStudio up and running, I would say close it and then do the following. Find the RMD file, in this case 08, the 8th lecture of the series. There were 4 yesterday, 4 today. We're number 8 right now. So it's 08 clustering hands-on.rmd, and I'm going to double-click on that and start RStudio. There's a reason for doing it that way. Maximize. Okay. So one of the reasons for doing it that way is that, oh, I hate this. Let's go down here. One of the problems we have is often, where is the stuff, and is the console synchronized with what knitter would think? If you start RStudio by double-clicking on an RMD file, it starts in that directory. So if the data that you want is in the same directory as your script that you click on, you won't have to go hunt around where it is. So we're going to do that. And we're going to start working through this script. Now, for those of you who are too lazy to work through it, there's an HTML file produced by knitting this. But I'm actually going to just work through it here and see what happens. Right? We have the title stuff in YAML. We have some cute stuff that you can ignore to make some of the HTML come out nice. We're going to look at, eventually, a breast cancer data set. Same breast cancer data set we've been looking at in some of the other examples. I'll continue with this example tomorrow morning when I talk about classification. So if you've got this stuff in the hands-on folder, by then it'll be easier to follow through. And as with the data that Greg and Maciej were looking at, it's originally on the scale of counts. I'm going to log transform it. And so I'm going to take log of something plus the data. And I'm going to choose the something a little more cleverly. It's an idea to, oh, by the way, I don't know if we taught you this in RStudio. There's these little buttons here. See the one that I've highlighted that is the green triangle pointing to the right? That means run this chunk. So I can click there, and it will run that chunk, and it presumably did something. Can't quite tell. I'm also going to fix something. I like chunk output in console. By default, it will try to put it in the same script up here. I like it to put it down in the console with figures going into the plot window. So let me rerun this and see that. So there it is. I defined something called epsilon, whatever that is. And I'm going to run it. And it's going to produce a plot. What this is the idea is, suppose I had data in 2 groups where one of them was twice the value of the other. That's because there are 2 biological magic numbers. One of those is 3, which is the default number of replicates for every biological experiment. And the other is 2, which is the fold change in expression for something to be significant. None of them, of course, actually mean anything. They're just everybody's favorite starting point. But we want to be able to detect twofold. And the problem is that if you add a little constant to something, you attenuate your estimate of the fold change. Do I want to write this? Yeah, I'll go over there and write this. And so the poor guy with the camera has to actually follow me for a moment. It would be nice if we actually had a dark enough marker. Okay. So if epsilon is 0 or close to 0, you look at the ratio of 2x plus epsilon to x plus epsilon. It's close to 2. But as epsilon gets bigger, So epsilon goes to infinity. This doesn't matter, and it goes to 1. So as you make epsilon a little bit bigger, you attenuate your estimate of this. It becomes less than 2. How does it change? Like that. That's what that plot shows, right? As you add bigger epsilons, your estimate of the fold change shrinks. So you can pick this so that if I wanted, for example, to make sure that, I don't know, I had an estimate of 1.98 whenever it was really 2, then I would pick something that way. So it turns out that on the breast cancer data, I'm going to execute the next chunk, 286 is the median data. So that's sort of the middle value there across this. And if I, what's the next chunk? Next chunk. Okay. And if I try that with that median value, if I want a 1.9, I can add up to 30 and not do it. If I just want 1.95 instead of twofold, I could add a 15. If I want a 1.99, I want to add something around 3. So I'm going to add something around 3 when I actually do something with this data. Okay. Now here's a big chunk of R code in a structure that I like because this is actually a time-consuming step. I'm going to read in the raw data, and I'm going to turn it into a format I can use in R. And then I'm going to save it in a binary R file, which is a more efficient way to keep the data and load it in. But I only want to do that once. So the R code says, if this file exists, just read it. If not, go through all these other steps. Read in the TCGA data on breast cancer, form it into matrices, log transform it, save the bunch. Since I've told you to download that, when I do this, it'll be relatively fast. It's loading in the breast cancer data done. If it was doing all that other stuff, it would take considerably longer. Okay? So we can start actually doing, oops. Okay. So what are we going to do? Well, actually, we're going to do some clustering both with and without filtering the genes. So we're going to try to get an idea of how to filter them. First thing we're going to do is on the log scale, so we've now log transformed the data by taking log of x plus 3. Okay? We're going to compute the mean and the standard deviation of each gene. That's what this does. I don't care that this was on a different version of R. Let's ignore that. And then I'm going to compute the coefficient of variation. How many don't know what the coefficient of variation is? Okay, everybody knows it, so I'm not going to explain it. If you don't raise your hand, you don't respond, I get to assume that you know things. All right, so we compute the coefficient of variation, and we look at a histogram. Okay? So for most things, right, the coefficient of variation is somewhere here around 0.1, like 10%. That's about how much varies. Some of them go up to about 40%. Some of them go up to 100%. Probably the ones at 100% we don't want to use. Those are like way too variable to believe that we have any reliable measurements of anything. Okay? So that's one way to think about how we might want to filter things. There's another observation that's worth making. This is related to a bunch of the things that Greg Rempala was talking about with borrowing strength. This is a plot of the standard deviation per gene on the log scale as a function of the mean per gene on the log scale. And so you just look at the blue stuff. I used a function called smooth scatter because otherwise it will try to put 40,000 distinct points on this. Smooth scatter does that basically as a density plot in 2 dimensions. But you can fit a smooth curve to that. That's that orange curve. There's a clear shape to how the standard deviation varies with the mean. So I used a method in R called LOS to figure out what that curve is. And then I added a multiple of that curve, completely arbitrarily chosen multiple. That's that red curve. I think I chose, 1.7. Why 1.7? Because I tried various ones and I looked at it and said, I like this one. Okay? And I also said, well, there's a whole bunch of stuff down near 0 that's probably crap. So I drew a vertical line and said, I don't want that really low expressing stuff. So that's some cutoff on the mean. And I don't want stuff below the red curve, because that's sort of the expected variation from the data as opposed to interesting variation. So I'm going to pick the stuff in the upper right portion of this, cut off by the red curves, as the genes that have enough variability and a big enough mean to probably measure something that's worth considering as part of the biology. And if I do that, so I've summed them, there's 1,025 genes in that upper quadrant. And I can draw another picture to show you clearly what they are. That's what's left. Is the orange stuff is what I'm keeping, and the stuff that's just blue in the background we're going to ignore. Okay? This has nothing to do with any outcomes. Those are the 1,000 most interesting genes, basically, in the data set. Okay? So that's often a preliminary to doing clustering. We're going to use my favorite class discovery package. And here we're going to do hierarchical clustering to get our first picture of the data. Okay? So we're using Pearson correlation and Ward's linkage, which again is my suggestion for the default the first time you go through. Okay? It is still running, so I have to fill some time until we get a result. Okay. So that's what clustering the samples look like. Notice that there's 1,003 breast cancer samples here. There's a lot of structure. How many groups of breast cancer patients should I believe based on this dendrogram? I mean, this is a question that's perfectly good to answer by democracy. You could just vote, right? There is no right or wrong answer here. Right? There's different ways to try to guess the answer. Right? Some people who would be very conservative with a view to the data says, I might believe 2 clusters. Clearly, I split it up there. There's that one group that's way off, not like the others. Other people might say, I don't know, 5 clusters. Right? If I cut it down at about 0.2, there's 1, 2, 3, 4, 5 clusters. So I don't know, someplace between 2 and 5 seems like plausible answers. Probably not going to believe much more than 5. Want to know how many of those are real? And this is using all the genes in the data. We could instead just use my filtered data, and that's what we'll do in this next one. Doesn't take quite as long to cluster because I don't have as big a data set. How many groups are there? Come on, give me some feedback here. So somebody thinks 5. Somebody thinks 3, right? Maybe 5. Maybe 7 if you really want to push down a little bit. Maybe just 2. Certainly not 1. Someplace between 2 and 7. We'll be really generous here. Okay? How do we tell which of those are real? This is a real biological data set. There's nothing made up about this. The data are the data. It is what it is. So we're trying to figure out what we can learn from it. Okay? Well, one of the things you could ask is, when I clustered with all genes, do I get the same answers when I clustered with the subset of 1,000 genes that have the most variability? What do you think? Well, one of the ways to do that is with the table command. And so what I've done here, I can cut into 6 groups each. So I cut the all genes filtering and the all genes clustering and the filtered genes clustering into 6 groups each, and just counting how many things get the same labels in that. And group 1 looks pretty good, right? Everything that got group 1 in the whole genome clustering, well, only 3 of them got any other group there, and only 9 of them got anything. So group 1, that's reproducible. That's the same whether I use all genes or just the 1,000 genes. Group 2, well, there's at least 2 groups here that it splits into, and it sort of smears across a bunch of groups there. What that tells me is if I try to make this into 6 groups, I get different answers depending on whether I use all genes or the filtered set of genes. So that's a problem. So maybe there's fewer groups than that. So I could look at 4, and well, again, this is that same group 1, which is really good. And well, this looks like a good group this way more or less, but it clearly splits up that way. I get different answers, even with 4. If I do it with just 2, well, 2, I get the same answer. So I can reliably say there are 2 groups. I'm not sure at this point whether I can reliably say there are more than that. Now let me tell you something about breast cancer. I know there are at least 3 kinds of breast cancer because some breast cancer patients have an amplification of the gene HER2. These patients respond when treated with Herceptin, which is a drug that targets HER2. And patients who don't have an amplified HER2 gene don't respond. They're really different. I know that because they respond differently to a treatment. Some breast cancer patients have overexpression of either the estrogen receptor or the progesterone receptor. If I treat those patients with tamoxifen, they tend to do really well. Patients who don't have that don't have a favorable response. I know there's that group. And then there's a group, which is the cleverly named triple negative. They don't have ER or PR or HER2, that don't respond to Herceptin, don't respond to tamoxifen, and so you give them some nasty chemotherapy because we don't have anything else to do yet. That's at least 3 groups. I know that. So 2 cannot be the right answer. Right? There's like real data. Okay? Maybe. Yeah. So that's part of it. So far, this preliminary look says 2 is the answer. But we know it's breast cancer, and it's all kinds of breast cancer. And I just told you that there's at least 3 groups. So we're going to try to pursue this a little bit further and see what happens. As I said, we have this notion of the silhouette width. Question?
How do you even know that those 3 subtypes are even present in your data set?
That's a good question. Part of the answer is this is an unselected cohort of breast cancer patients. So this was an all-comers kind of study. So we at least strongly suspect that they're there. We're going to actually look further and see that they're actually there. But that's an excellent question. Right? If somebody had just picked out triple negatives to study, we wouldn't see the others. But I'll show you later that they're really here. Okay? Anybody else have another question? Okay, so as I said, we have this tool that claims to help answer the question of how many clusters, known as the silhouette width. This again is that block where I don't want to run that if I don't have to, so I put the answers, the pre-computed answers there so I could do it quickly and load them in. And this next block actually shows you what the silhouette widths are. So with 2 groups, it's a 0.31, and then it drops to 0.14, 0.12, 0.10, 10. Clearly, the silhouette width strongly votes for 2 groups. Okay? Which we've already said is the wrong answer. And there's another way to look at it. You can count for each time that you split it into groups and look at the silhouette width. You can ask, how many samples does it think are in the wrong group? When you do it with 2, it thinks 40 are in the wrong group. And then it jumps up to 142, 155, then in the 200s out of 1,000. So it thinks that if you try to do 4, 5, 6, there's a couple of hundred out of 1,000 samples misclassified. So it doesn't think that you know how to classify things using hierarchical clustering. Okay. Now the other thing that I told you about is the bootstrap. Again, this takes a while to do, so we're going to get the saved answers and see what we can tell about what these look like. So there's the silhouette width plot when there are 2 groups. Group 1, everybody's right. Group 2, there's just a little bit that it thinks are wrong. Okay? We'll work through these. The just plotting the bootstrap plots takes a little bit of while because there's actually some computation that's going on in the background before it figures out what the heat map is. And it is, after all, 1,000 by 1,000 heat map, seeing where each of those pairs of 1,000 samples goes. I would really like a picture to come up. I'm trying to keep track of time to see if I can actually, there we go. Okay. There are at least two groups. There's no question about it. Okay? That first split, that one group that went off by itself, is really different from the others. Okay? Why am I not getting anything? Can we see my next plot? I hope. Okay. So there's the silhouette width for 3 groups. Okay, I mean, there's a few more guys that it thinks are wrong. This one will take a little while. I probably won't do all of the bootstrap plots. I may actually switch to the HTML where they're pre-computed. We'll fill in some here. Because what we're interested in is whether the bootstrap gives at least some evidence for at least 3 clusters instead of 2. The silhouette width doesn't. Okay. Okay. What do you think? 2 groups or 3 groups? This shows pretty strong evidence for a 3rd group. It's small, all right? But this is that same group we had to begin with. This one looks like another group that we can split off. Okay? Let's see. How do I get back to... Okay, I don't know how to get to my HTML. I guess I'm just going to have to go through this. Let me try to look at one more of these. See whether there's some evidence of 4 groups. All of these figures are pre-computed. You can get the HTML version and look at it. We'll do that at some point. I want to at least wait to see whether there's some evidence of 4 groups. And then we're going to see what else we can say. I don't know how to tap dance, so I don't know how to keep you amused while we're waiting for the next figure to come up. Okay? Okay. What do we think? Are there 4 groups? Maybe. Right? I mean, there's some evidence. Here's our first group, which is labeled red up there. Here's the second group we found, which is labeled in purple. Here's a 3rd group that maybe blurs into the other one. And here's sort of a core of a 4th group. Looks like there's some overlap between those, but there's some reason to believe there might be 4 groups. Okay? You can go down and try 5, 6, and 7 and convince yourself that there's clearly not 7. And we can instead try to build a heat map. And the way we're going to do that is, we've already clustered the samples, so we don't have to do the clustering of the samples again, but to build a heat map, we have to cluster the genes. This is what the 1,000 genes look like. We said that we should scale things for purposes of display. We should truncate, but to figure out where to truncate, we maybe want to look at a histogram of the scaled values. And I decided that about 2.5 standard deviations looked good to me. Okay, that's where the red lines are. So that's where we're going to truncate. So we truncate it. And now we make the heat map, which hopefully won't take forever, to see what we get. That's a start. I'd like to see the rest of the figure. It does still have my little red stop sign there to stop it if I was going to interrupt. So it's still trying to do something. I like RStudio for lots of things. My feeling is that for complex image plots, it tends to be slow, which you're probably not going to argue with me given the example here. OK, so I don't see the dendrograms. I don't know why that's the case. I may try zooming it to see that. But if I look at the heat map, my red group is the one that's easy to tell from everybody else. Okay? And you can see that there's this big green block here that is not, well, it's not present here or here. Some of it's in here, but it's sort of mixed with some red stuff. Right? So there's a bunch of genes that are low in this red group that aren't really low most of the other places. We can clearly tell this purple spot, we see another sort of green stripe there. So that purple group is, again, different from the others. It's the blue and the green that are hard to tell apart, right? Finally, the dendrograms. OK? All right, there's this block of genes here that maybe tells part of the blue and green groups apart. There's a red block in there. That's some genes that for a subset of the blue group are different. So we think there maybe really are 4 groups. OK? Because there's some evidence here, some structure that does that. Again, the strongest structure is we can pick off the red piece. Then we can pick off the purple piece, and then maybe the blues and greens. So we're going to try to look at this another way to try to tell if we believe yet that there are 4 groups. We're going to use those colors for samples as we plot other stuff. Okay? So we're going to do principal components. So we fit the principal components model. And this is what's known as a scree plot. Anybody know what scree is? So the evidence is that the statistician who named this was probably a mountain climber, because scree is the loose rocks that fall at the bottom of a cliff, so that sort of heap of stuff down at the bottom of a cliff. This is the scree, and that's the cliff. OK? What's being shown here is the percentage of variance explained by each component. And since the first one explains the most, it has to be biggest. They have to go down. So we can certainly see that the first 2 have to at least be significant, maybe 3 and 4. But after that, it just begins to look like crumbled cliff parts forming scree at the bottom. So there's somewhere around 2 to 4 components. That's important in terms of how many we want to look at and how we think this matters. So there's a plot of the data in the first 2 principal components, colored by the clusters we found from hierarchical clustering. One of the things that we see is that the first principal component by itself is doing a good job of spreading these things out. The red group is off to the left, clearly different from the others. The purple group is in the middle. We can pretty much tell that. Maybe overlaps a little bit. The blue and the green are to the right, and those are the ones that have the most overlap. This is the same picture that we got, in effect, out of looking at the bootstraps, that we could tell the red and the purple reasonably well, the blue and the green overlap. Okay? But now we have a 2-dimensional picture of it that actually reflects real distance as opposed to where you are in the dendrogram. Okay? Now, what's nice about this is that we can color this in different ways. So we're going to take this same plot, and this goes to your question. How do I know these groups are here? And what we're going to do is we're going to color it based on the expression of the estrogen receptor, which every normal human being thinks has the name ER. It's not the name that it has according to the Human Gene Nomenclature Committee, which is the official group charged with giving genes names that confuse the rest of the universe. Okay? So it's actually called ESR1. Okay? That's the official symbol, which is what's used in this. And so we're going to split that at this, we'll see why at some point, 9.5. Bigger than 9.5 is going to be colored magenta. Less than 9.5 is going to be colored cyan. So we will click the do this button and see what happens. So that first component is basically the expression of the estrogen receptor. Okay? This is high expression of the estrogen receptor. In most cases, this is low expression of the estrogen receptor, except for a few outliers thrown in there. Okay? So that says at least some of these groups are here. The estrogen receptor can be high or low, and it's reflected in the clustering. I probably should have colored this with HER2/neu, but I'm actually going to plot, there's a which 3, 4. I'm going to plot components 3 and 4 and color them by HER2/neu. So this isn't perfect, but the third principal component is at least partially related to the expression of HER2. Now, the actual physical thing that tends to go on, well, it's partly expression, but it's actually a DNA measurement. When I say that HER2 is amplified, it means we have many more copies of the part of the DNA that has HER2. But we actually see that there's, in the third component, there's sort of the green group that's low and the purple group that's high. So that's where we're going to stop, because that's as much as I got prepared here. We could think about what we want to do with this. Right? My take on this right now is that there's at least 3 groups. There might be 4. Right? One of those groups is, turns out, we can't quite see it from this because I didn't, maybe we could do the right plot here. Let's, since we have actual code here. Let's copy that and plot the first 2 components by HER2/neu, which I should have put in here, and so we'll do it now. Okay. So remember that this stuff, which was red in my arbitrary colorings, was low in expression of the estrogen receptor, and it's low in expression of HER2. That says that's triple negatives. Well, I didn't show the progesterone receptor, but that's the guess at that. That red group that's most different from the others is likely to be the triple negative breast cancers. Okay. There aren't enough different colors in the universe because I reused purple, right? But if I go up to my original plot, which was here, right? That purple group, so we need to think about what that one is, that is low in the estrogen receptor but high in HER2. So the purple group of samples is likely to be the HER2 amplified. Okay? And so all of those blues and greens are not HER2 amplified and not triple negative, so they're all hormone receptor positive. So the real question here biologically at this point is that 1 or 2 different kinds of estrogen receptor positive samples? And can we tell how reliable that split is? Now, the other thing that we know about breast cancer is that people have been trying to figure out subtypes of breast cancer using high-throughput technologies since at least 2001. Chuck Peru at the University of North Carolina, who worked with Pat Brown at Stanford, who developed some of the first 2-color microarrays, has published a series of papers doing this that started off by saying there were 4 kinds of breast cancer, triple negative, HER2 amplified, and what he called luminal A and luminal B, which is 2 different flavors of hormone receptor positive. He's also published papers where he thinks there might be 5. Some people maybe think 6. But early on, in that very first set of papers, he claimed from microarray gene expression data that there were 2 different subtypes of estrogen receptor positive breast cancer. There's some evidence of that because you can sort of see that there's overlap between the blues and greens, but they're sort of core samples that clearly are far enough apart that you might think they're subtypes. Or you might argue that there's some continuum there, and we could explore that. That's what we're going to look at in the hands-on tomorrow when we talk about classification. The challenge we're going to look at is, can we reliably tell the blue ones from the green ones? So, any questions?
What is the biological meaning of variability? The expression makes sense.
That's an excellent question to which I'm not sure I know the answer. What it sometimes means, right? What it means sort of in part of this, right, is that if we think about this first dimension as estrogen receptor expression, which we think is what it's associated with, right? We know there's a group that's high and a group that's low. So if I look at the variability of ER, it's high because there's a big difference between it. So when I measure that variability, sometimes it's an indication that there are multiple subtypes based on the expression of that one gene. Another thing it can mean in the context of cancer is that if you've got some pathway that gets broken in different ways, and you sort of look at the expression of key genes in that pathway, they could be both much higher and much lower than normal. They could just be more variable because they're no longer under the sharp control that they normally are. Okay? Any other questions? Okay then. We'll see you tomorrow.